Good afternoon, everybody. My name's Carolyn Hutter. I'm the Division Director for the Division of Genome Sciences at NHGRI, and I want to welcome everybody to today's WALS lecture. First, a couple of housekeeping notes. I want to thank the WALS team, particularly Diana Gomez, for organizing today. If you didn't pick it up on your way in, on your way out, you can pick up handouts about upcoming WALS speakers. It's a very impressive lineup. I also want to thank my colleague, Lisa Chadwick, who nominated Dr. Cox today to come. Lisa is a remote worker, so she's watching remotely, but I wanted to thank her for her work in the nomination and in organizing the day. If you are wanting CME, the CME code is 50111 up here on the screen. And for when we get to the Q&A, people in the room, I'm going to encourage to use one of the two mics, but we'll also have a large number of people participating online today. And so online people, to submit a question for the speaker, use the Send Live Feedback button that you'll find on the videocast page that you are watching this on. So in terms of introduction, Dr. Nancy Cox, a professor of medicine and director of the Division of Genetic Medicine within the Department of Medicine at Vanderbilt University Medical Center, a lot of medicine in that title, and founding director of the Vanderbilt Genetics Institute. She had training at Yale, Washington University, and the University of Pennsylvania before going to the University of Chicago, where she was a distinguished faculty in the Biological Sciences Division, and then moving to Vanderbilt in 2015. She has an active research program focused on integration of functional genomic information to aid in discovery and interpretation of associations. And she's done pioneering work in translation of genetics and genomics through use of the Vanderbilt DNA Biobank, BioVU. I could go on for the whole hour with her numerous honors, but I'll just call out that she's a co-winner of the 2008 American Association of Cancer Research Landon Award, winner of the 2010 Leadership Award in Genetic Epidemiology, and recently the 2023 American Society of Human Genetics Leadership Award. In addition to her research, I just personally want to note that she has a very strong focus on training, mentoring, and workforce development, and she's been an inspiration to many early career geneticists, particularly women, seeking increasing leadership roles in the field. She's certainly been an inspiration to me. In addition to that, she has many active research and advisory roles in genomic consortia. That's where I currently interact with her the most, and this includes her being a member of the NHGRI National Advisory Council for Human Genome Research. So it's truly an honor to welcome Nancy Cox to give today's WALS lecture on prioritizing genetics to reduce existing health disparities. Nancy?
Thanks so much, Caroline, for that generous introduction. And it's such a pleasure to be here at NIH. It's such a storied place for research in the United States. And I was a frequent visitor to the NIH campus for many, many years. And we've kind of gotten away from that over time. There's a lot more security on the campus now, the primary campus now. Many of our NIH-sponsored meetings here in the Bethesda area end up being at satellite hotels or at satellite NIH buildings further out. But it's nice to be on campus again. I was here for many, many kinds of meetings over many years, and also, know many intramural scientists here. So it's been a real pleasure to catch up on all the science still being done here that is so important and worthwhile. So I'm really happy to be here talking about this topic, which has taken over my life. And so I'm really going to talk about how much more heritability lab values used in everyday medicine are than physicians realize, and how has led to really institutionalized health disparities that can't be fixed without genetics. And part of this is that bringing genetics into medicine is creating new kinds of health disparities. And we all know that. There are important new health disparities in both the common variant space with things like polygenic risk scores for risk prediction, but also in the rare variant space with how much harder it can be to interpret the genomes for anyone from whose genetic ancestries are really outside Europe. In contrast, a lot of the laboratory values that we use in everyday medicine are trying to capture primarily dynamic changes that happen as we depart from healthy homeostasis into either a disease state or something that our medications are driving us in the wrong way to have. But the problem is that these laboratory values are more heritable than physicians realize, and especially the ones that are old and more general indicators of health than specific indicators of particular disease. The genetics is often completely non-predictive of the outcomes we're using the measurements for. And so when those laboratory measurements are highly heritable and that genetics is not predictive of the outcomes, the genetics is not just misleading us, but it's creating a barrier to the dynamic part of the measurement telling us the right things about where people are in the distribution. This makes it an opportunity to use genetics to improve health and equity, both, not by using the genetics to change risk prediction, by regressing out the genetics of places where it's misleading us. So all this work was done in the context of the biobank at Vanderbilt University, where we have EHR data and going back an average of 10 to 15 years and more than 30 years in some subjects. We have more than 300,000 samples, DNA samples in the biobank now, and we have genome interrogation on about half of those subjects in various ways, GWAS, exome sequencing, whole genome sequencing, but now there's an alliance that is going to be sequencing the entire biobank, so a pharma alliance is sequencing the entire biobank, which will really allow us to do a better integration of the common and rare variant space. But we've always also used the genome variation to impute other omics into the data. I thought 10 years ago, as we were doing more multi-omics studies, and these are biomarkers that are so rich. All of the multi-omics data is very, very rich for predicting future health status. That those things would make their way into medicine as biomarkers that we would use commonly in thinking about health and disease. But everything changes very slowly in medicine, and for sure, transcriptomes, metabolomes, proteomes are not nearly as hardened in terms of interrogation as genomes have been, and we need to move in that direction in order to be even thinking about using these in a clinical context. But the biomarkers that we use very routinely are often telling us much, much more about very late stage departures from healthy homeostasis. And I think the opportunity of the omics measures is to be catching departures from healthy homeostasis potentially much earlier. One of the things I hadn't appreciated till I got to Vanderbilt is that while the part of this that has the genomic interrogation is a fantastic resource, the part that doesn't have any genomic interrogation. The 2.8 million other people with just 15 to 30 years of electronic health records data is a fantastic additional resource for enriching what we learn from the genetics, but also for a big sample that can lead to discoveries that we are able to take into genetics to validate. And I think as we think more about using genetics to do target development for better therapeutics, the other things that we can do with data like this are thinking about the pleiotropic effects of genetic factors earlier in life so that we use biological information to think about surrogate endpoints that are not Alzheimer's because it's so late in onset, but about some of the other biology driven by the same genetic factors that are driving at least pieces of the Alzheimer's genetic puzzle. And I think as we learn more and more, think about the big contribution that biology attributable to microglia, which are just macrophages, might be telling us about much earlier in the lifespan that we could profitably use in thinking about clever drug designs to not wait until people have cognitive decline to be thinking about doing therapy, but rather looking at the sort of same kinds of failures in other classes of macrophages that may be manifesting earlier. But everybody fully understands that introducing genomes into medicine has led and is continuing to lead to new kinds of health disparities. And part of it is totally not in our control except as citizens to do anything about. This just shows the increase in income inequality in the U.S. from 1967 to 2014. And in fact, these trajectories got worse after 2014, and it was really only over COVID that there was some decrease in income inequality that reflected investments that were made to try to keep people healthy and safe during the midst of the COVID outbreaks. That inequality of access is then coupled to historical failures to have the right data from diverse populations to invest in with genetic studies. It makes sense to use data that are very rich and have a long history of information, clinical information and medication information to use in investing in the first genetic studies. But because those studies had much less diverse data, less diverse with respect to representation by gender, less diverse with respect to representation of the diversity of the human population, that we have substantially oversampled European genomes relative to genomes in the rest of the world. And the problem is the very success of the science in leading to discoveries that have some good opportunities for clinical translation means that populations that can afford to have more genetic information used in their health care continue to do that. So even though we are now collecting much more genetic data on diverse populations than we ever have, it's accumulating faster, it's an impressive worldwide effort, we are still accumulating information on the genomes of Europeans even faster. I don't see a way to catch up until everybody has genomes done routinely for health care. I really don't think we will catch up in terms of the representation of world populations in interrogated genomes until that happens, because even worldwide there is inequality of access to information and inequality in the ability to invest in the research enterprises to create all of the data that would really have value for interpreting. So we know that we can't move forward without creating new kinds of health disparities. And so this makes it the case even now that in N-of-1 studies in children and adults from diverse populations, we're less likely to get an unequivocal solve. We're more likely to be dealing with variants of unknown significance. And that will be the case for some time yet. And this is a worldwide problem, and we are, I mean, everybody is trying to invest in ways that improve these outcomes, but it will take time, and it will be hard to catch up with the quality of information. In the common variant space, polygenic risk scores are being delivered to diverse populations even now, but the truth is, what we need to learn from polygenic risk scores, what we have any opportunity to deliver, involves a lot of tail properties. Like most of what is happening in the middle of this distribution doesn't matter. I mean, for understanding risk, you're at the same level of risk as the general population. What we generally try to deliver is more information from the tails, and it can be the high tails of risk. We need to understand low risk as well, especially as we're able to better combine rare and common variants in a way that really helps define accurately large-scale genetic risk to disease. But tail properties of normal distributions are the least stable part, right? And different ways of calculating polygenic risk scores have the most impact in the tails of the distribution. So small variations in how calculations are done in the populations in which these predictive models are trained have a big impact in who ends up in the tail that's going to get information that they're at high risk. That's not attractive from the statistical perspective, but it's made even worse by the fact that most of our methods for both calculating the original the training of these risk score predictions, but also the application, ignores the reality that about half of the U.S. population is continentally admixed in recent generations, and that's the fastest growing part of our population, all of which is ignored at some risk of failing to capture important aspects of population level risk fully accurately. And so the failure to have had enough genomes early on to ensure that methods accommodated the level of complexity that we need to deal with in treating populations in the U.S., in delivering genetic data to populations in the U.S., is going to haunt us for some time. We need to improve methods that are already being used to deliver results to our populations. And so we can hardly do this fast enough because it's already happening. So we know by translating genomic discoveries, we are creating new kinds of health disparities. But we have an opportunity in the lab test space to use genetics in a way that will improve health and health equity. And we use laboratory tests every day in medicine. And the failure to have fully appreciated how diverse human populations are in these measurements has created a number of really important institutionalized health disparities within medical centers that I think really have to be addressed. So in our center at Vanderbilt, in the Vanderbilt Genetics Institute, Leah Davids led the effort to clean the laboratory values in a way that it could be shown that we were estimating the same levels of heritability, getting the same GWAS signals as studies done on those laboratory measures in a research context. The reason it takes so long and is so hard to clean these values is over time, we go to different vendors for getting laboratory tests run. Over time, they change the units in which laboratory values get reported. There's, massive batch effects that can vary by places where the tests get run. And all of that, you want to try to standardize to make sure that we capture the essence of the genetics that are really underlying these biomarkers, but they definitely succeeded in that. You'd show at the end of this process that they published and then created really this pipeline that's being used by lots of medical centers now to clean and standardize their laboratory values for further genetic studies. But you can get the same estimates of heritability. You see the same top GWAS signals. We're really capturing sort of the heart of the polygenic architecture of laboratory values in the hospital setting, in this clinical setting. So what proportion of the lab values that physicians order and interpret on at least a monthly basis, or we might say even that we have data on at least 1,000 people in our biobank for, what proportion of those lab values do you think are 10% heritable? I ask these questions in 20%. Yeah, so if I ask, or even what proportion of lab values would be significantly heritable in that context, in a group of physicians in a grand rounds, if I say how many people think 10% of labs you order would be significantly heritable, a lot of physicians will raise their hand, and 20% and half the hands go down, and 30% and they'll say, yeah, then if my husband's in the audience, he's the only one still raising his hand because he remembers I'm a geneticist and I wouldn't be asking otherwise. But no, it's 2/3 of laboratory values that we have that many people on that are ordered at least once a month are significantly heritable in the EHR. A substantial fraction of those, more than 30%, between 30 and 40% of laboratory values are significantly heritable, have heritabilities in excess of 40%, and up into the 80, 90% range. And why is this a problem? Because physicians think of them as really dynamic measures of disease risk and progression, and that's how they are used, as primarily dynamic measures of what's happening right now in this system. And the genetics is static. The genetics is anchoring people at some part of that distribution and trying as hard as possible to keep them there. And that's why it's actually a drag on the dynamic part of these measures. Certainly, there could be some possibility that any gene by environment interaction, any exposures that are really changing these values could be interacting with the genetics. But for the most part, the genetics are static. And because they're much more genetic than physicians are often giving them credit for, that means that first time somebody orders a creatinine that looks like it's putting people into stage 2 kidney disease, they're just watching to see that progress because they think that person is in stage 2 kidney disease, but the genetics may be misplacing them in the distribution. They may be at stage 4 kidney disease. They may be perfectly healthy. Creatinine is 60% heritable, and only a fraction of that heritability has anything to do with kidney function, as I'll show you in a minute. So then, if you think about these being heritable and more heritable than physicians realize, What proportion of the heritable lab values have mean values or variances that differ significantly by EHR-reported race ethnicity? 10%? A quarter of them? Half of them. Half of them have significant mean differences by EHR-reported race ethnicity, and a not insubstantial fraction differ significantly in the variances. And especially among populations of recent African genetic ancestries, the fact that there's a much longer population history, larger effective population size, means those populations have much more genetic variation. They have much more functional variation. It is not uncommon to observe laboratory values with significant mean differences where the African Americans in our biobank are more likely to be outside reference ranges, of course, in the direction where the mean has changed, but actually are more likely to be outside reference ranges on both ends of the distribution, despite the fact that there's a big mean difference. This is, in some ways, this is a crazy thing, but the truth is many of the reference ranges were set a very long time ago in what we would now think of as ridiculously small samples of white men. Some of the lab commonly used measurements have undergone changes over time with respect to reference ranges. Most have not. Most of them are not adequately corrected for sex differences in the measures or for how diverse these laboratory values are across populations. So I'm going to go through a few examples of studies that have been done on commonly used lab values. White cell count, neutrophil counts, commonly used to, in people who take drugs that might be cytotoxic, in trying to identify people who might have hematological disease. The Duffy polymorphism is a well-known, completely benign polymorphism that substantially lowers white cell counts in people homozygous for this polymorphism, most of whom have recent genetic ancestry from Africa because it is an adaptation to high malaria environments that is extremely effective. Allele frequencies in environments where this particular adaptation is effective, allele frequencies are nearly 1 for this variant. Nearly everyone is homozygous for this variant in those parts of Africa. So on average, African Americans in the U.S. the allele frequency is 0.8. That's about the proportion you would expect given the average admixture of European alleles in recent African ancestry groups in the United States. That means 2/3 of African Americans are homozygous for this allele. And when a bone marrow biopsy is ordered in a patient homozygous for the Duffy allele, and the only reason for the bone marrow biopsy is low white cell count, it's like never positive. There's no return in the investment of that bone marrow biopsy. It is never positive. And that was shown at Vanderbilt and a group of other biobanks. So there's unnecessary bone marrow biopsies still being done, despite the fact that this benign ethnic neutropenia is supposed to be well known to physicians. Bone marrow biopsies are still being ordered, not uncommonly, in medical centers where the only indication is a low white cell count, no additional symptoms. And when those people are Duffy homozygotes, there's no return on that white cell count. On that bone marrow biopsy. But there's a lot of drugs that we give that can be cytotoxic. Physicians look at the first white cell count that happens after patients get put on these drugs, and they are taking patients who are Duffy homozygotes off first-line meds for autoimmune conditions, for cancer chemotherapies, drugs people take after transplantation. They're taking Duffy homozygotes off their meds when they don't need to be. African Americans who are Duffy homozygotes, people who are Duffy homozygotes, don't have fewer white cells. There are fewer in circulation. They're hanging out in peripheral tissues, and as soon as there's an infection, they come out, they fight that infection just fine. The low counts of white cells in circulation are not indicative of an increased risk for infection. So taking people off these important first-line drugs is disadvantaging them in their health care. And again, you see this really dramatically for drug withdrawals for hemopoietic toxicity, but not for other reasons that have to do with the inability to just take the drug, other kinds of side effects entirely. So it's all about Duffy in people taken off the drugs for hemopoietic toxicity. And on average, they get to take about 20% of the first-line meds that other African Americans take when they have lupus. And we've always known that people who have lupus with recent African ancestry do worse with lupus than we see with other populations. But how much of that is taking a substantial, two-thirds of African Americans are at risk of being taken off those first-line meds very early. So as I say, Duffy's a benign polymorphism, except for how it undermines healthcare in the homozygotes who carry it. It is not a genetic health disparity. It is something else. Should never lead to a health disparity, but it leads to a lot of repeat testing, unnecessary bone marrow biopsies, undertreatment in lupus that I just showed you. But under the hood, the threshold for white cell count to get into cancer chemotherapy trials was for many years going to keep half of all African Americans out of clinical trials, because two-thirds of them who are Duffy homozygotes end up falling below the thresholds all the time. And they set those thresholds because they don't want to lose people in the course of clinical trials who are already starting to have cytotoxicity from the treatments they've had. But of course, when they're Duffy homozygotes, they're going to be lower. It was an inappropriate threshold for African Americans. And while that's being changed now, that's part of the reason why African Americans were so underrepresented in chemotherapy trials for so many years. What it's doing at the level of transplant, people are looking at, but it's not anything good. I can tell you that now. How bad it is, we are still looking at. But you can see it even in the rates at which African Americans ended up having burst appendices, because many of the rubrics for when you do surgery, when you should do surgery to keep the appendix from bursting, required reaching a certain high white cell count that people who are Duffy homozygotes will not get to. They'll be right at other populations' averages when their white cell counts have really gotten dangerously high. And as a consequence, for some period of time when those algorithms were in common use, African Americans just had more serious health disparities reflecting burst appendix. Race-based reference ranges cannot solve this problem. 1/3 of African Americans are not Duffy homozygotes. The distribution of their white cell counts looks like everybody else's distribution of white cell counts. It's not about the social construct of race. It's about what alleles you have, and common alleles with big effects drive huge heritabilities. The heritability of white cell counts in populations of recent African ancestry is much higher than the heritability of white cell counts in other populations, because, in part, of this allele that has a big impact on average cell count and is very common in that population. Most alleles that have big effect sizes are rare. This is an example of how selection has driven an allele to very high frequency that's benign for health, but not benign when we think of how it influences health care. So what do you even call this disparity? It's not a genetic health disparity. It's a kind of ignorance health disparity. But it's not one that can be fixed without using the genetics. White cell counts are useful measurements. We should be recognizing, and these are studies that are about to be published, the polygenic background for white cell counts is not predictive for any of the outcomes that we use it. It's not predictive of who will develop infections when they take medications. Just the pure polygenic background, even in European populations. What's valuable about white cell counts, neutrophil counts in these contexts is that dynamic nature. The way genetics influences where you are in the distribution is noise for how we use that measure, and that measure will be more informative if we regress all the genetics out for everybody. It will be a better, more effective biomarker for the ways that we use it. Hemoglobin A1C is an important measure for understanding how well the control for blood glucose levels is in people with diabetes. It is now not infrequently used as a way of diagnosing diabetes in healthcare systems. But people who carry G6PD alleles, mutations, again, that happen to be adaptations to malaria environments. This is a very common adaptation that has arisen over and over again, independently, at different alleles at G6PD, which is on the X chromosome. So males are homozygous for these alleles. These alleles have arisen all over the world where malaria is endemic, and they're not rare in those populations. Collectively, G6PD mutations that can have this effect on hemoglobin A1C range from 5 to 20% collectively in populations where malaria is currently endemic or has been in the past, because we carry around those variants for a long time after the pathogens are no longer driving them. The reason that this affects hemoglobin A1C levels is that there's red cell hemolysis, which affects the average lifespan of the red cell. What we're doing is measuring the glycation of hemoglobin on red cells. And in people without G6PD variants leading to red cell hemolysis, we're getting a snapshot of the average circulating glucose levels in about the past 3 months, which is the rate of red cell turnover. In people with G6PD variants, there's more rapid turnover of red cells because of the hemolysis, and you're looking at something closer to a month or a month and a half of life of that hemoglobin molecule in its glycation. But when you don't know who's a carrier and who's not, you can't take that into account. And on average, in people with diabetes, those carrying G6PD alleles have about a 1 point lower hemoglobin A1C level for males, and you can see the progression in females. Even the heterozygotes have a somewhat lower level, and then homozygotes more similar to what we see in the male homozygotes. So what happens as a consequence of this? Certainly people with diabetes, seeing their physicians on a regular basis and having this measured, are not being treated as aggressively because their physicians and they themselves think they are doing better at controlling their blood glucose levels than they actually are, a full point lower on average in males. This means in our biobank, on average, carriers of G6PD alleles have a 1.2-fold increased risk in any diabetic complication, which is highly significant. It's a particularly serious thing for diabetic retinopathy, which is one of the sentinel complications in diabetes. In early clinical trials to understand how well blood glucose control would reduce the risk of complications, diabetic retinopathy was the sentinel one because it's often one of the first of the diabetic complications to arise. So hugely significant, 1.8-fold increased risk in diabetic retinopathy. Highly significant even in our biobank, but I can tell you biobanks all over the world are seeing this too. We're working in Primed in a big consortium to really look at this in a serious way. It's the same everywhere, and we have good information from Singapore and Taiwan, but also the UK, because these alleles are really common all throughout Mediterranean populations where malaria has been endemic until recently and still is endemic in some parts of Southern Europe. So here's an example of polymorphism that has nothing intrinsically to do with diabetes. It affects this measurement of glycated hemoglobin that is a relatively inexpensive and effective assay. In populations that have the kind of healthcare system where hemoglobin A1C is tested every year to identify diabetics, it's an average 7-year later diagnosis of the diabetes in the G6PD carriers. So it's not just that they're not being treated aggressively enough, but if you use hemoglobin A1C to diagnose the diabetes, they'll have it for many years before you recognize and treat it. The cost of diabetic complications is one of the reasons why the National Institutes of Digestive Disorders and Kidney Disease exists. Diabetes is a huge public health care problem exclusively because of the complications that it causes and the expense and difficulty of treating those complications and the morbidity and mortality that arise because of those complications. We have always recognized that minority populations have more diabetic complications. And it's easy to say, well, they can access health care less. They have less access to things like glucose meters that give continuous feedback on how well they're doing. But part of it are these kinds of things. And removing this one health disparity using genetics would pay for the genetics for everything else. I mean, it's hugely consequential in cost to have people go blind. And as I say, these alleles are not rare. And on this particular biomarker, they have a big effect. They have effects on other biomarkers too, because of the red cell hemolysis. Blood-borne pathogens, the adaptations to blood-borne pathogens are going to have big effects on a lot of things we measure in blood. We have ignored those things in interpreting laboratory values in ways that are truly disadvantaging a lot of the world's populations. And we can fix this, and we would improve healthcare in general and health equity if we do. I'm just going to go fast through the kidney story because, I mean, this is just a crazy way that geneticists and nephrologists have talked past each other for a long time. You can make direct measurements of kidney function through glomerular filtration, but in general, we estimate glomerular filtration using these common biomarkers, creatinine or cystatin C, that are filtered through kidneys routinely. One's a metabolite, one's a protein. And the equations for using this to estimate glomerular filtration really just take sex and age into account, as if the variability in circulating levels of these biomarkers was only attributable to kidney function. The heritability of cystatin C has been estimated in the range of 70 to 80%. And a huge proportion of that heritability is attributable to the structural locus for cystatin C genes on chromosome 20, where there's a huge effect of EQTLs. So there's a big genetic influence on where people are going to sit in that distribution for cystatin C. Creatinine is just polygenic. There's not big effect alleles, but it's 60% heritable. The only part of the actual genetic variation in that space that's telling us about kidney function is the part that's shared between the two of these. They have nothing intrinsic to do with kidney function. They're just cheap and easily measured proteins or metabolites that get filtered through kidneys, and using them as if the only thing affecting their levels is kidney function is selling short what the biology of these things actually is. They have their own genetic reasons for being present in circulation, and affecting kidney function isn't one of them. They're just convenient ways that we assay kidney function. The magnitude of genetic effects, so this is UK Biobank data, cystatin C levels, huge effects, and we took out the chromosome 20 region because we wanted to keep cystatin C and creatinine on the same scale, and the p-value for the cystatin C structural locus is like 10 to the minus 5,000. It's such a big effect. And of course, they're identical, whether you use raw cystatin C or raw creatinine and the estimated level, because it's just a linear equation. So the genetics is the same for the raw biomarker as the estimated glomerular filtration. And when I talk about this with nephrologists, the first thing they say is, well, no, but we only care about the changes. That's the only thing that matters. It's the changes that tell us. Except many people with their first creatinine level are in some stage of kidney failure. So we're using absolute values, not just changes, in the way we label people and follow them. And somebody said to be at stage 2 kidney disease may be perfectly healthy or may be at stage 4 just from the distance the genetics that have nothing to do with kidney function takes them. So it leads to the heritability can lead to misstaging. This contributes to what we see as failure to progress, but it also is something that is probably, based on our data, would be sometimes mislabeled as acute kidney injury that leads people to early death. These two biomarkers really have nothing in common in terms of what they do, except that they're both recycled through kidneys. So the shared genetic architecture is a way to understand kidney function. And the thing is, they both have, they have some important shared biology. So the top is serum creatinine, the bottom's the cystatin C. But they also have absolutely unique big effects. And, of course, the cystatin C structural locus on chromosome 20 is a huge effect present in cystatin C that's not in creatinine. We could estimate GFR better by regressing out the genetic parts that are not about kidney function. We'd have a better measure of the truly dynamic part of what's happening when kidneys start to fail. And so, you could do a PRS that's just about that shared genetic architecture that really is telling you about the likelihood of future kidney failure. But you could do an EGFR, estimated glomerular filtrate versus cystatinine, removing everything that isn't shared with across these biomarkers. And so, you would just remove all the heritability and use the genetics as genetics. Or you could do these other kinds of regressions that focus in on just the dynamic part that matters. I'm going to stop talking about this now so we can have questions. But sex is a problem too. And women have been disadvantaged in liver transplant for a long time because the sex differences in measures of liver function were never taken appropriately into account. There are no simple solutions to complex problems, and this whole business of the heritability of commonly used laboratory values is a tip of the iceberg problem. As I said, half of these have population differences in average values, half of the heritable measures, and most of the measures are heritable. If we even focus in on just the highly heritable ones that have important population differences, we've got dozens more important biomarkers that we have to address. And you just can't go forward on the same path once you know about this. When you know about harms that you are doing every day in medicine, we have to figure out how we can fix these as fast as possible. And so because we've used genetically predicted gene expression so much, we're really used to the idea of separating out the genetic component from all of the other things that influence gene expression. And I think as geneticists, we're focused on the vase very heavily. But we can use the genetics to see much better the consequences of things like environmental exposures and social determinants of health and learn that biology. And when the genetics is noise for how we use a laboratory value in medicine, we shouldn't hesitate to remove it. We do a lot of things with laboratory values under the hood. So what comes back from the laboratory that's ordered often goes through some equation before that is submitted to the physician to interpret. All we'd be doing is regressing out the genetics in that same kind of equation. So you could think about this as individualizing reference ranges by using the genetics, regressing out the genetics through changes in the individual reference ranges that are provided to physicians. And our physician scientists think that is the best way to do it because it would help physicians internalize how diverse their populations actually are with respect to these measures. They would learn and understand more about genetic diversity in laboratory measurements. But I got to tell you, physicians who have 9 or 10 clinics a week hear that and they're like, are you nuts? I don't want to see 1 million different reference ranges for my patients. No, if you think this is destroying my ability to interpret laboratory values, take all that genetics out, blow the distribution back up to what I expect, and let me use the reference ranges that I'm familiar with and know how to deal with. So I think we need to do a study to really look at physicians and think about how we try to fix this. But it has to be done under the hood. It has to be done in a way that doesn't require physicians to know and understand the genetics of Duffy in order to interpret a white cell count. And here's a recent publication that shows the distribution of white cell counts in Duffy homozygotes relative to the means for the population at large. So it's a big effect of a single allele on a commonly used biomarker. And I do want to shout out to my colleagues in the health disparities research that we do. So I've been working with Consuelo Wilkins since I first got to Vanderbilt. She was the first non-geneticist who came to me to ask me to collaborate in studies that she was doing. And she always tells a funny story that she had to work up the courage to say to me, while we were planning our first grant, Nancy, I don't really think genetics have very much to do with health disparities. I was like, oh, yeah, me either. I mean, they're mostly about other things, but we don't want to leave on the table anything that we could fix using genetics. And so that's been our mantra through now 2 generations of studies. And then our EPIC PRS group, that's part of the PRIME consortium, we were all over this in this grant, and they really have thought of very clever ways of trying to get at some of these things. And it's been a real pleasure to work both in that consortium and in this group and my funding. And happy to take questions that I thought I would leave more time for. But I do get carried away with this stuff and really apologize. I should have left more time for questions. So while we're waiting for her to find questions from online, are there any questions from the audience?
I was going to say, we do have a few minutes. Is the microphone working? Please come to the microphone. And for people who are here in person, there'll be a reception right after this, so just in 5 minutes, so we can also do some questions then. And people online, please feel free to submit your questions, and we'll see what we can do. So go ahead.
Thank you very much, Nancy. It was a great talk. I am Leonardo Mariño from NIMHD. And I would like to ask you, do you think that having reference values for diverse populations or for, let's say, different genders and different times in different times, different ages will do as opposed to what we're using right now before we have to get to the point that you're talking about?
Actually,
where
no.
You have...
I'm uneasy that that's a one-size-fits-all kind of solution that won't fit anybody. And I mean that sincerely for the substantial fraction of laboratory values that are heritable. The idea of reference ranges that try to use just population-level information is going to be, at best, misleading and mostly wrong for everybody. And
is it going to be better than what we have right now?
I doubt it. I mean, because I'll say, we used to use race in the creatinine calculation of estimated glomerular filtration. And that was not better because it was not about the actual biology. It disadvantaged populations of recent African ancestries in really prominent ways that almost certainly kept people who needed kidney transplants from getting them. Taking race out of that equation doesn't remove the big mean differences that are there. People are still disadvantaged by the genetics, but they're not disadvantaged in a particularly race-based way. They're disadvantaged by the genetics that we fail to adequately accommodate in the way we do the calculations. I think that more individualized reference ranges for things like sex and age make a lot of sense. But I'm uneasy about population-based reference ranges that would capitalize on allele frequency differences among populations, because those really don't work for any individual. You have to know the alleles. And so I think would they be better? in most cases, I'm skeptical they would be better. Maybe for things like age and sex, when those have a prominent role in the interpretation of the biomarker. But if it's, it's really just about age and really just about gender, yeah, we should be doing things like that.
Okay, thank you.
So
we don't.
So we've hit the 3 o'clock mark. Are you coming down for a question? So we'll unfortunately have to have this be our last question, but again, availability in the for the reception outside. Go ahead.
I want to thank you for a beautiful talk. I've been trying here at NIH for the past 2 years to get people here to get themselves a whole genome sequence, including all the RNA, etc., and expression. And you have your examples, you're using a lot of euphemisms in your examples, but basically it comes down to you have to diagnose first and treat second, not the other way around. And so just thank you again for your talk.
Thank you. So happy to answer questions outside. And if anybody listening in has questions that they didn't get to ask or think of later, I'm happy for them to email, and we'll definitely respond.
So wonderful. So let's thank Dr. Cox again. And I will note to the side, Nancy, if you can point for me, we have a Walls plaque, which we will be mailing to her, not making her carry in her carry home. But thank you again for everyone who came in. Please pick up the list of upcoming Walls. I was looking at it, very impressive lineup on your way out, and please join us for the reception. And everybody participating online, we really appreciate your joining us this afternoon. Thank you.